in what we have to present today. I'm going to start off with about a 20 to 25 minute talk, and then we'll invite Dr. Ian Walsh, our guest here, who will talk a little bit more about the East Coast in Florida. And then we'll open it up for questions. But if I could ask you to save your questions to the end, and we'll have a discussion together, okay? All right. So I want to acknowledge a few people. There's two people here that basically keep the instruments going. And that's AJ and Jeff. And Rick provided some insight and some pictures and slides for me today. So thanks to you all. So this is kind of an idealized cartoon of the coast and large-scale processes that we're going to be talking about today. Basically, when it rains on peninsular Florida, all that rainfall comes down, trickles into the rivers, it goes over agricultural land or urban runoff, roads, whatever, and ends up in the estuary, in the lower estuary, around Sanibel in the coastal zone, where it can drive production of phytoplankton, which are tiny microscopic algae that grow in the water column. And it feeds a lot of the food web that's here. However, when you get excessive nutrients or one particular nutrient that's normally limiting, you can cause blooms of algae, and that leads to other problems like hypoxia and causes some of the animals to leave the area and is actually not as harmful to the ecosystem. So. This is something that maybe you all are familiar with, but I just want to explain the concept. This is from a review, but basically 1950 to 1990, they described the input of especially nitrogen to the coastal zone, but we've really done a good job after World War II of creating a lot more nitrogen than there was historically to grow crops and so on. So it's a lot of excess nitrogen in the environment now. And what's happened is, especially in the coastal area, you start to see early indicators like some phytoplankton blooms or filamentous algae, and at some point it leads to these excessive blooms. So the title of my talk had to do with El Nino. We've had kind of a very exceptional year this year. Started in 2015 and carried through until today. Where we have what's called an El Nino, and I just want to describe what that is. So in the Pacific, you have very strong trade winds blowing from east to west. When those trade winds start to weaken, warm water flows up and basically blocks a normal process from happening there called upwelling, and it changes the jet stream from being one that's higher in North America to one that dips down low. So you end up getting flooding in a lot of the south. And this year we noticed that in particular we had a lot of rainfall. So this is from one of our sensors. We look at rainfall as a major driver. This year, in January, starting in January, we had 10 inches of rain, and a lot of that rain fell in a few big storms, cold fronts that came through, and that wasn't just on Sanibel. That was all over peninsular South Florida. So we had a lot of rain. Normally we get around 2 inches in January. So those of you who are here, you probably know that South Florida is relatively flat and has a flood control component to it because of some history there. And Lake Okeechobee is kind of the center point for where we store water and when we have excess water, where we have these high volume discharges. You can see just that there's a connection to the Caloosahatchee, there's a connection to the St. Lucie, and then there's a water that flows from the north. There's some water that goes south, and because of some policy changes and flexibility in the last 2 years, there has been more water that goes south, but a majority of it actually comes to the west coast and leads to excessive discharges. So this is just a plot of the level of Lake Okeechobee. The red is a historical average, and then where it's exceeding that historical average, what's happening is the lake fills up faster than they can get rid of it, and there's a big problem with that because it can the dike is old and they're trying to fix it, but you can have flooding, and there are a lot of lives and people that live in the area that could be affected by a catastrophic flood. So they start discharging, and this year they discharged really high volumes right when these two sort of periods of time when there was a lot of water coming into the lake and they weren't able to get a lot out, so they started pushing it to the coast.
Excuse me, are you going to make the slides available?
Yes, we're going to make the whole presentation available online, yeah. So this is just a plot of the flows. The black line is kind of our harm threshold that is recognized by a lot of science done by various federal and state agencies. And it was a necessity because of flood control that we discharged a lot of water. And some of that water is from rainfall falling on our watershed. So we had excessive rain, plus the lake was filling up, and it led to these really high discharges. And they happened twice. Once in February, and then we sort of got to a situation where things were drying out, but then the normal wet season started again, and the lake started filling up. So and it started raining, so we had another big pulse of water in the beginning of June. So our perspective on this comes from data that we collect hourly at a bunch of places throughout this large area, and I'm going to try and integrate a few different sites for you to tell the story of what happened since basically January. And I'm going to talk about this site here, which is out in the Gulf of Mexico. This is Sterile Island, Fort Myers Beach. I'm going to talk about McIntyre Creek quite a lot today, which is in the National Wildlife Refuge, and then we'll also show some data from up in Fort Myers. But all of these sites have the same equipment. It's underwater. It records every hour, sends data, and it sends a variety of parameters, and they're listed here. Salinity is one of the parameters we're really interested in, but we're also interested in dissolved oxygen and chlorophyll. And chlorophyll is an indicator of how much algae there is in the water, okay? So there's a great way of plotting the data. It's all available to you if you want to look at it. You can do all these plots yourself. But I'm going to first show this period from basically January till March, that initial pulse that occurred during the dry period. And you can see salinities in the Gulf of Mexico, we're getting down to 20 to 25, which historically, the green, well, I don't know if you can tell these colors, but the green is this year. So salinities are very low compared to average or normal years. The other years that are shown here, the 4 years previous, are all pretty much around 32 parts per thousand, which is like full strength seawater. So we're getting these departures way out in the Gulf of Mexico that are low salinity, and it's pretty far away from where the estuary is because of the high flows. So just to make some comparison, because I'm going to talk about the site again, is McIntyre Creek. This is in the National Wildlife Refuge. It's a recent report that analyzed a lot of hourly data like this and showed that a lot of the variation that's occurring in salinity is because of the discharges at the Franklin Lock, which are both watershed and lake discharges. And this is another site, the blue line again. Now we're really getting low salinities. This is 0 to 25. So every tidal cycle, and this is a place, Shell Point, where the Caloosahatchee is fairly constricted still. It's acting more like a river. But every high tide, you're getting some salt water. So imagine if you're an animal that likes say 20 parts per thousand to 25 parts per thousand, trying to survive in an environment that's going from 0 to 30 every day, twice a day. So it's a really hard place to make a living. So one of the things that caused me to want to share some of this information with you today, I started plotting the data when I found out Ian was going to be visiting. And I looked at the chlorophyll response, which is basically, again, this phytoplankton, this bloom-forming algae that was in McIntyre Creek is the green, and the Gulf of Mexico is the red. After that big pulse in January, you see this bloom occur. And after thinking about it, it was probably a diatom bloom. It wasn't necessarily harmful. Some of this is food for filter feeders like oysters and scallops. But it can lead to high oxygen demand, hypoxia, and other problems in the ecosystem. Well, as I expanded my view to the summer, it's really interesting. So here's the first pulse of water. Here's the second pulse. You can see at McIntyre Creek this sort of 2-week delay in terms of the peak flow versus the peak of the algae. So that's called a lag. So it's like a 2-week lag, and that makes sense because it takes about 10 days for water to get down into the estuary, get mixed, and then be put out into the Gulf of Mexico. What was also interesting is the second phytoplankton or diatom bloom that occurred here, most likely diatom, we didn't actually look, it's chlorophyll. It occurred in McIntyre Creek, but not in the Gulf of Mexico. That suggests that the water that was trapped in Pine Island Sound in San Carlos Bay was had a high enough residence time, which translates to it allowed the bloom to get going and really grow, whereas in the Gulf of Mexico it was being diluted or transported elsewhere. So when you sort of say, well, why did this happen, we can look back at some previous work that's been done here, and it shows a really good relationship between nutrient loading, okay, which is what Frank and I were talking about earlier, and chlorophyll abundance. So, you know, it's known what the cause is, and it was just to show you the conclusions by the scientists at the time. It basically showed that this is the case, and it says restricting the loading of the limiting nutrients should control eutrophication. So I guess that's my message for that particular, I think, extraordinary year and a couple of algae blooms that were occurring in the lower estuary. So this is kind of a widely known thing among scientists, and if you look here, there's a great series of pictures. One's Lake Okeechobee, which I thought was interesting, this green cyanobacterial bloom, which I'll talk about in a second, and then a green macroalgae bloom that occurred in China. And they also agree that reductions in nitrogen and phosphorus are what we need to do. So, but I did want to contrast those events with some other things that I've seen and that Rick has documented well. It's probably hard to see in the back, but this is an image taken from space of Lake Okeechobee, and there's algae. It's a huge, enormous cyanobacterial bloom, or a blue-green algae bloom, that occurred starting in May. Some pictures of that downstream of the Franklin Lock. You can see just how visible it is, and Ian's going to talk a little bit more about the nature of the cyanobacterial bloom and how it manifested itself on the East Coast. But we were able to document it. We were able to see it and count it, and also in June the county went out and observed different places where it was occurring. It usually stops where it gets salty because they like fresh water, sort of stagnant water. So if you looked at our real-time data, you can see it's this increase that starts on May 1st and then goes back down. I know it's a little bit subtle, but look at the sort of disconnect, if you will, between the two big pulses. It's not like as easy to relate to those pulses by occurring in the middle, and we sort of have some idea of what's going on. But if you looked at another pigment during this period, so this is May 1st here, July, you can see by looking at phycoerythrin, which is specific to blue-green algae, there was quite a lot of it around the upper estuary. And we know because we watched it on TV, right, for the last 2 months, and it played out. But blue-green algae, as Ian will talk a little bit more about this, but it tends to outcompete diatoms and other algae when phosphate is abundant, okay? So not so much in the example I showed before, those blooms were most likely nitrogen limited. They tend to thrive in warm temperatures with stagnant water, and they clump and float. So it actually makes it really hard to detect in situ the way that we're doing it in the middle of the water column. It does produce toxins, but we don't really understand when and where it produces those toxins. So there's a great site that Lee County staff and others have put together recently that has great fact sheets about blue-green algae as a result of this summer. So I encourage you to look there. And the other that really interesting thing about microcystis is that it has gas vacuoles, so it's able to regulate its own buoyancy. So it'll come to the surface and take all the light and basically exclude other light need requiring plants from growing. So it's for being a primordial, very ancient organism, it's quite smart. So the last phenomenon, I guess, or algae bloom that I wanted to bring up is lately we've seen, and this has really been in July, late June and early July, we've been seeing green macroalgae on the beach. And sometimes it's quite extensive. Sometimes it's been observed just growing on individual sand grains in the swash zone. And this is an ulva. It's a species that's fairly tolerant of lower salinities. Most macroalgae are pretty much like really salt, well, not salty, but ocean strength seawater. And this one, it can tolerate brackish water at times. And it really loves nutrients. And there's been quite a lot of work done on it because it's an indicator of nutrient pollution. And more recently, like this week, we've been seeing a lot of sea hares washing up. Sea hares love to eat macroalgae. So we think what's going on is that the boundary of the estuary has been extended. There's algae that's growing because of higher nutrients, both from the discharges and percolating through Sanibel because we've had a lot of rain. It's being pushed out through groundwater. And then these little sea hares are marching along and following the abundant food along the beach and being washed up. Now, they're washing up for unknown reasons, but it's possible that there are small pockets of hypoxia because of the high oxygen demand that I talked about a second ago, and then they're being stunned and then being washed up on the beach. It's really hard to know why they're showing up in such high abundances. So this again is a phenomenon that's pretty well known among scientists. You have this sort of pristine state with seagrass, and then you have a eutrophied state, or a state where there's lots of abundant green algae and it's using plenty of nutrients in the water. So just to tee up Ian a little bit, I did want to mention the Tealight compounds in the water, the brown substances that really caused a lot of the angst among businesses that were seeing this brown water in front of their hotels and others. I mean, all of us really. It's from their Tealight compounds. They're a visual indicator of fresh water. So the more dark it is, the more fresh or brackish the water is. Mostly, it's not harmful. It's not harmful to people, but it can block light that is needed for submerged aquatic plants. So I just wanted to mention that because I know there's been a lot of questions about the dark water. So my summary is that we've had 3 different types of algae blooms in this extraordinary year. And, you know, it's rainfall driven, but watershed and flood control policy that's led to these high flows. I think we had this diatom or diatom-like bloom in the lower estuary, and the blue-green algae bloom and the macroalgae bloom. And I just wanted to end there, and we'll like to invite up Ian. He's going to say a few words about what they've been finding out from similar sensors on the East Coast.
Thanks, Eric. Well, great to see a lot of people here. We had a lot of people here on Wednesday. Well, over at Harbor Branch on Wednesday. I'm down from Oregon and started work on this, I think, July 5th or so, and have been basically spent a couple of weeks doing this. So it was a great relief to give the talk over there. And then Eric and I said, "Hey, Eric, I'm in the neighborhood. I'll come over," forgetting that it's 3 hours across. Didn't seem that long the last time, but I was coming from Miami. This work is the Lobo systems that, to a large extent, because the Lobo systems were established here, Harbor Branch got them to monitor the systems in the St. Lucie and Indian River Lagoon. And AJ and Eric have been great helps to get them started going, and I hope they'll continue to work together quite a bit. Systems are slightly different. They're much more nutrient oriented on the east side, so they've got the nitrate and the phosphate sensors over there, along with the chlorophyll, CDOM, salinity sensors. So we're going to go through a fair amount of data. I'm going to go relatively fast. I gave this talk about 45 minutes over there, so I'm going to compress it. So I was told at the end of the talk by one of the cameramen, well, I didn't get any of the data, but I figured out what you were saying. So I hope we get to that state. And then I just want to acknowledge Harbor Branch scientists Brian LaPointe, Dennis Hanasak, and Kristen Davis, who essentially, Kristen is essentially the AJ over there. She's running the system, keeps it going. And I should note that when I told people I was coming over here, everyone said, "Say hi to AJ." Nobody said, "Say hi to Eric." And their data is also available on the website, fau.loboviz.com, and it is a fantastic resource to have really high quality data delivered to you seconds after it's collected. I mean, what's the lag time? 5 minutes maybe at most? 10 minutes? So people like, what's your name? Anthony, who's volunteering here this summer, they're going to use this data through their career. We used to put instruments out and go collect them and hope that we got data. Now you see it right away. And the other advantage to that is AJ and his team know when to go out and fix the problems because they see the data. And that's just an amazing ability. Okay, so now that I've spent way more time on the intro, then I'll have to catch up. Pea soup, guacamole, whatever. It showed up there, and it made, I think I saw this on BBC and Gizmodo, so when it gets into the tech news, you know that there's a weird and wonderful situation going on. All right, so we're going to look at the IRLON sites, Indian River Lagoon Observing Network on the St. Lucie Estuary. They have more sites up on the Indian River, but we're going to stick with the St. Lucie. We're going to look at the biogeochemical data and sources. So Eric gave a great introduction. A lot of that is the freshwater and seawater mix and how that moves through the estuary. That's the critical part of the entire estuary. And then looking at the biogeochemical data, we're going to look at the dynamics of the system in harmful algal blooms. So really I started doing this because I took a look at the data and I didn't understand it. It didn't make any sense relative to what was being reported as what was causing the bloom. And so I called Brian LaPointe and I said, okay, I'm missing something here because what's out there and what I'm seeing don't match. And the great thing about having live data is you just follow the data, and you can figure out what's happening from the data itself. You can then, you should have a knowledge of what the systems work, but the data will tell you what's happening. And he said, no, you're right, and that's a message that's just not getting through. So we'll go into what that is. That's the Lobovis site for Florida Atlantic Harbor Branch. So here's the sites that we're going to look at. The South Fork site here, this is next to a control gate. So the C-44 canal goes off this way. That's where the Lake Okeechobee discharge comes in. The South Fork 1 site, or South Fork site here. This is the Roosevelt Bridge, where I think at least one of those pictures that I showed a couple of slides was taken. The North Fork site, and then this is the urbanization area, high urbanization area. Middle estuary, and then closer to the ocean. This site shows up in a couple of the graphs, but we're not going to talk too much about it. Okay, so just to reiterate again, here's our two sources. Big water coming in and out of Lake Okeechobee, and of course, ultimate source is the seawater. And these are the sites on this map. Now there are canals coming in from the west and from the northwest. We're not looking at those data in particular, but I'm recommending that we should be looking at it more in the future. All right, here's our discharge data from Station 308. We're looking May to July. So between basically the end of May through the 4th of July holiday, when this is where it really started to pick up in terms of press coverage, etc., they were running about 1,800 cubic feet per second through that whole period. Then, in response probably to the outcry, it went down quite a bit and then came right back up again. So that was a cool slide that you showed of the overall and the flood control. I mean, obviously you want to get rid of the water and not lose the dam. It's really not a question of why you want to do it. It's a question of how and when. All right, so we're going to take a look at the estuary flow traces just to follow the water around. So our two are basically the CDOM from the pherometer on the LOBO systems, and as Eric said, that's looking at decayed organic matter. And then the other one is ocean water. We follow that with the salinity. Both of these are relatively conservative within the estuary that we're looking at and over the time scale that we're looking at. So here's the first data. This is, again, the data all from the sites. This is all of the data from January through May. And I was expecting sort of two end members, seawater and Lake Okeechobee. So at seawater, this is salinity on this axis, CDOM on that axis. So seawater has very little dissolved organic matter in it, and obviously lots of salt. Lake Okeechobee, very little salt, and a fair amount of CDOM. I don't know what your CDOM levels are around here, range. Similar range? But it shows up actually as a nice triangle here, and here's a very high, moderately brackish water with very high CDOM loads. And this is runoff and other freshwater sources. So to a large extent, that's the water coming in from the north and from urban runoff. Just to carry that message home, this is January data. Again, 10 inches plus rain. Normal is 1.5 to 2, so a lot of water. So this is just the January data, and now we see there's really not much of an input from in the estuary here. From Lake Okeechobee, it's pretty much all the water is either seawater or the fresh water from runoff.
For those of us who've never looked at a trough before, could you just talk a little bit about more of what we're seeing on this?
Oh, sure.
What is a scatter diagram it looks like?
Sure. Yeah, so it's at any given point, it's the two sensors readings from that hour at that point. So this data set is coming from this site. This data set right here is coming from this site. And then the triangles are, okay, so now I'm colorblind, so I have to go look for the shapes. So that's here, that's the North Fork. South Fork, I think, just gets, or the middle estuary just gets buried, and then the red squares here are the South Fork.
The time is not an axis, though.
Time is not an axis other than I've taken all the data from January. So January, lots of rain, lots of runoff, Lake Okeechobee was not being discharged, and all the water in the estuary is somewhere in this mixing. So if if you're mixing a black and tan, you end up with something in between. If you're doing an Arnold Palmer, you end up with something in between. You can think of those as your end members here. This was June, July, up until I think the 7th or so. Note, there's no connection now with the seawater. These are our sites here, and this is the 2 sites in the Indian River Lagoon. So we've lost the runoff end member. There's probably a little bit of here, but basically we're looking at Lake Okeechobee water entering and very little brackish water formation. So when the discharges were high at these sites, particularly these 3 sites, we don't see much of the runoff signal. So it changed the estuary quite a bit. So those 10 plus days of 1,800 cubic feet per second dramatically changed the estuarine dynamics within the system. And we're going to go through that quite a bit here. All right, so that's CDOM and salinity. The other way to look at this now is CDOM versus phosphate. So the nutrients are coming mainly from the runoff here. So here's our seawater end member down here, and it's not pure seawater because it's already in the lagoon, but there's very little CDOM and there's very little phosphate. The ocean doesn't have much phosphate. Here's our runoff end member up here at the North Fork. Very high CDOM levels and 6 to 7 micromolar of phosphate, which is really high. This is the South Fork 2 site right here. So this is sort of our Lake Okeechobee water entering the estuary. CDOM is about in the middle at 60. Phosphate's about 1. So the headlines that said loading of nutrients from Lake Okeechobee is driving the bloom isn't true, because it's the nutrients up here are really already there in the estuary. The nutrients coming in aren't terribly much higher than, they're significantly higher than 0, but they're not hugely higher compared to the North Fork and going out. So that's when I called Brian when I plotted this up. I said, this doesn't jive with what's in the papers. Okay, so then just to reiterate that, our ocean water, very salty, fairly low CDOM, and very low phosphate. The Lake Okeechobee water is not salty at all. It's fresh water. It's got a moderate amount of CDOM, and phosphate's around 1 micromolar. Now, 1 micromolar is enough to drive a lot of plant growth still. And I'm not talking about the nitrate right now, but we're going to get into that in a moment. But that's still significant to grow plants and phytoplankton. But our runoff end member that's coming up from the North Fork of the estuary, brackish water, 10 PSU, very high quinine sulfate, and very high phosphate. It's greater than 6. I picked a relatively low number. This is data that we're getting as we looked at it, updated hourly, which is actually sort of interesting trying to do a talk, because you do your figures and then you go back and now 2 days later there's new data. Oh, now I got to redo all the figures, but it's a fascinating process. So when I called Brian and said, this is what I found, he goes, oh yeah, well, he wrote a paper that was published in 2012 from data collected in 2004 and 2005. So a 6 to 7 year lag between collecting the data and writing the data. Most people, you don't really do actions on the basis of 6-year-old data, but he's doing it for a scientific process, understanding the system. You still want to understand the system. It's great for science. It's hard to manage on the basis of 6-year-old data. He also did a paper in 2015 and in 2016 a study for the local counties. But all of that pretty much confirmed what I had been saying too. He's just got more data and more in-depth analysis. But the Lobo systems with live data can lead you to those same conclusions, but in a real-time basis. And so for a management tool, for looking at what the flows are, looking for the harms, or even to the extent that I'm sure the charter boat people, fishermen use the data from the network here, where am I going to go today to fish? Or if I'm scuba diving, where do I want to see? Am I going to be in low visibility, high visibility? I want to see the groupers. I don't want to just be seeing sand particles. Okay, so a little bit about the nutrients. So we're looking at both the phosphate and the nitrate, and Eric introduced this very, very nicely. We look at the ratio between the two, and that's called the redfield ratio. If you take phytoplankton, general phytoplankton, and grind them up, you'll see that the atom ratio in the material is about 16 nitrogen atoms for every phosphate atom. So the redfield ratio, named after Redfield, who discovered this, 16 atoms of nitrogen for every atom of phosphorus. So in a not accident of history, seawater actually also tends to go to the 16-to-1 ratio. So normal oceanic water that's nutrient replete and the phytoplankton that live in that water both have the same ratio. When they get out of whack, you get more specialty phytoplankton. And when phosphate is abundant relative to nitrate, so that rather than a 16-to-1, it's 16-to-2 or 8-to-1, that's when you get the blue-green algae. And one of the ones that compete very well in warm fresh water is Microcystis, which actually has a microcystin toxin in it, which is pretty nasty. Not the worst of the HAB toxins, but pretty bad. Okay, so we're going to look at each of the sites, and I'll run through these very quickly. What I did was I took the nutrient data from the Lobos. I plotted phosphate on the left, nitrate on the right, and where the phosphate in there, so this is 0 to 8, this is 0 to 100 and whatever 16 times 8 is. And so this is redfield ratio graphs. So where the phosphate is above the nitrate, it's replete in phosphate. That's where you're going to get microcystis growing. And when it's the opposite, you're going to end up with more diatoms. And so Eric showed that actually in his data sets, in which the different events had different phytoplankton. You had diatom blooms and microcystis blooms happening. So this is the entrance. This is the water coming in from Lake Okeechobee, and it's relatively low for the estuary, and relatively in redfield ratio. So it's not particularly phosphate enriched, and it's not particularly high in nutrients, within the range that we're going to see here. We come further into the estuary at the South Fork. Now we're seeing pretty much high phosphate loading all the time, and some curious spikes that we're going to get up to, we're going to look at later. But here now we're seeing higher phosphate than nitrate. We're above redfield ratio. Again, that's going to indicate microcystis are going to be very much happy in that environment. In the North Fork, now it's dramatically higher. So much higher phosphate than nitrate in the North Fork. And that's the runoff end member right here. As we go then towards the ocean, the middle estuary, which is carrying now water from both the North Fork and the South Fork. It's in between the two. We get to near the ocean, and we see some freshwater influence and high phosphate here, but by the time we get to June 27th, and that's a significant date, so remember that for the moment, June 27th, we're seeing pretty much just oceanic water here. Redfield ratio, very nicely, tightly bound, and beautiful tidal signal. All right, so the curious spikes in the South Fork Lobo. When we were talking before, high salinity means low phosphate. High salinity means low CDOM. But in this case, within those spikes, we actually have positive correlations. Salinity goes up, CDOM goes up, phosphate goes up. So that doesn't fit in our 3M member plot from before. So what's happening? So here's just the correlation between the phosphate and salinity through about the 25th, and then it goes away at about the 27th. Curious date again. 27th? Okay, so here's CDOM and salinity. And again, that pretty much goes away around the 27th. Well, here's our discharge again, the discharge slide again. And the 27th is pretty much close to the end of when the high discharge period into the estuary. So when we look then, so we're going to now look at this time period and then this time period. So previous to and up to the discharge going down, and then after the discharge went down. All right, so now this is hard for me to see, but here's our CDOM, here's our salinity, and they couple very, very nicely through the 7th, July 6th or so. So where salinity spiked, CDOM went up. Salinity spiked, CDOM goes up. After the 7th, now we start to see salinity going up, and CDOM really doesn't get much higher. So now we've got more of the source that we expect, which is more oceanic water. Salinity is rising, but there's not much phosphate in it, and there's not much CDOM in it. And so that's sort of normal. So before, as it ramped up, as the discharge ramped up, we started to see a fourth M member show up. But once the discharge went back down, then we start to see a much more normal flow. And that's, I think, really what's happening here. That's the key to what's happening in that, which led to the bloom, is that the source of water to the South Fork was actually the North Fork. So it wasn't flow from the ocean that was coming back into the South Fork. It was actually flow from the North Fork going into the South Fork. And the North Fork has high CDOM, is brackish, and has high in phosphate. So it fits all the criteria for where that water comes from. And that change in dynamics of the circulation changes with the discharge rate. So it's not that the Lake Okeechobee is adding more nutrients into the system. It's changing the estuarine circulation there. It's essentially creating a lake there, and the water is not flowing out. It's still flowing out, but it's flowing out relatively slower. Think of filling up a bathtub at some point. You're just, you've got enough going in, more coming in that's going out. You're going to raise that level coming into the South Fork. The South Fork kept going up. So let's look at the basin relationships to see what that works. So in normal, relatively normal flow, we're going to see the blue arrows, that's the oceanic water coming in. It's going to come all the way up into the South Fork and up into the North Fork. The flow out of Lake Okeechobee, these arrows, not really sure exactly what's going to happen here yet, but I'm putting them in. Some of the South Fork water is going to go into North Fork, but for the most part, the freshwater return flow is going out the estuary. Okay, and that's normal times. So now when we think about that, in terms of, now I'm plotting the discharge and the nutrient level, in this case the phosphate level. So here's our discharge going up. This is the South Fork data. I'm going to blast through these. Am I doing time? Ooh, doing okay. Discharge is going up, ramped up. Phosphate is going down. However, once they cut the discharge down, the phosphate came back up. So that's really cool. That means that water, high phosphate water, is coming back all the way up to SF2. So in a normal estuarine pattern, you're getting the oceanic water all the way back up to that gate. At SF, we see basically static at about 2 micromolar through here. Now this is the other slide that's nice. So here's our North Fork data. And notice that we've got nice tidal dynamics. I didn't connect the lines here, I should have, but there's a lot of data here. The one thing the Lobos do, give you lots of data, which is great for understanding things because you can test everything and think about how everything's working, especially on a very complex system, tidal systems, rainfall. There's lots of unknowns here. So getting more data allows you to test the hypotheses very quickly and sort through them very quickly. So up until, it's the 27th of June again. Tidal dynamics in the North Fork. between the 27th of June and through the low discharge period, we're really not seeing a tidal signal here. So our phosphate is high in the up to the 6s up here, goes down to about 3, and really not much of a tidal signal. And I think that what that says is that there's so much water, fresh water dumped in here, the tidal signal doesn't show up at all. It's just a big blob of fresh water moving around. All right, so let's test that. Here's the middle estuary. Now remember, before there was a big change between the middle estuary and the North Fork. Well, watch, I'm just going to click back and forth. Watch this time period versus this time period. Oops. There's not, I have 2 fingers doing 2 keys. That's really complicated. There's just not much change here relative to here. So it's basically the same water mass in the middle estuary and the North Fork post June 27th. And here's our estuary. Here's our near the ocean cut. And here's our tidal dynamics that's showing up. So phosphate, again, high loading in freshwater, low loading in oceanic water. And that shows up very nicely here. It's gone by the 27th of June. So that doesn't mean that the tides aren't coming in, but remember, we're fixed. The position of the instrument is fixed. Just means that the freshwater lens is way above that, okay, a meter maybe. But it's buoyant enough that it's not getting mixing. So here there's still tidal mixing, but by this time there's so much fresh water going out that it's not mixing down. It's buoyant enough and staying up. So the salt water is not coming in very far, and the fresh water is taking up all of the flow through that cut. So the discharge volume is changing the estuarine circulation. That's particularly happening between the North and the South Fork, which increases, and you talked about residence time, it's increasing the residence time. That means these plants double every day. It doesn't take too many days to create a real mess with lots of phytoplankton. All right, and again, this is Dennis Hanasek and his team showed this also in a couple of reports last year and earlier this year, and I grabbed 2 slides. Again, this is just the previous ones. For my own purposes, I grabbed the data down, which you can, and play with it in your own software. This is the LoboViz plotting. So here's our South Fork phosphate. Let me make sure I get this right. Here's our tidal. This is the pressure, so that it's essentially looking at the tidal, how deep the water is. And you see nice positive correlations between the pressure and the phosphate at the South Fork, which means that you're getting higher phosphate at high tide, not low tide. So low tide should be when the oceanic water is further away, and the fresh water is there. But instead, it's completely opposite. You're getting the high tide has higher phosphate. So when during this time period, which this is the 17th of June through July 5th, here's our June 27th, you've got an opposite relationship to what we'd said we'd expect. And even more dramatically at North Fork, higher phosphate at low tide, which is really weird. And the explanation for that one is we've just got this big ball of fresh water here, and the gradient in the North Fork is higher phosphate towards the north, we're pretty sure, and lower phosphate here. So that the tide is not moving oceanic water into this. Note this areas are very small. Again, these are all inferred. That bolus of water is moving back and forth here. So when on high tide, it's pushed back here, that's lower. That's actually the dilution effect of the Lake Okeechobee water. And then when it's on low tide, that bolus water moves this way. Now we get more North Fork water. So it's all about where you are relatively to the sources. And the 2 sources here and here and here all change. And that Lake Okeechobee water, instead of a normal estuarine circulation, has turned this area in particular into a lake. And if you remember, most of the reports from the very high bloom were right in this area. So, I think I said it about 18 times at the previous one, live data really is the key here. It changes the world from interpreting the water in the systems, and it's just an amazing change that's happened in my lifetime. Okay, discharges from Lake Okeechobee changes the estuarine circulation, which leads to higher residence times, and excess phosphate then leads to the microcystis blooms. Yep. Oh, so we came up, Brian and Dennis and I said, all right, so what do we really need to know more about this? One, we need to add the microcystis specific fluorometers to the system. We don't have that. We just got the chlorophyll. Two, we don't know what the nutrient levels are in the flow in the lake itself. This is not measured. So we're recommending to put a Lobo system on the lake side of 308 gate. And we also don't know the inputs from the north and the northwest and the west into this zone, the North Fork. So we're recommending a Lobo up here. And that'll constrain whether so that nutrient loading that's very high in the North Fork is coming from the farm and ranch lands to the north, or whether it's the septic systems and the urbanization in the area itself. And that's critical for how you attack the system going forward. Then build a flow model to make sure that you can change this. And I think probably the most interesting one, which is actually fascinating now, we recommend that they start modulating the discharge. Don't just drive it constant for 8 days, but go up and down on some cycle, using this model to make sure that they keep estuarine circulation going. So it turns out they've already started doing that. And right now the salinity level here is in the 7 to 8 range. And guess what? The HABs are going. So they are responding and they are changing it. But a model and more live data will allow them to use basically the salinity levels, the CDOM values from these 3 sites to modulate the flow and keep HABs from going. And I'm sure all of the people who live and work on the water would like that to happen in the future. And that's it. $257.
I know some of you approach me and have to leave, and that's fine. We'll open it up to either of us for questions or comments.
I had a question about on Ian's presentation. 2 things. Did you include rainfall in looking at some of those dynamics, because it can be so variable where the rain falls? And 2, is the circulation potentially influenced by sea level rise?
Yes and yes. I tend to put lots and lots of slides in, but that's the way I think, and then I cut them down.
Yeah.
So yes, we looked at rainfall through that period, and but just to simplify the discussion, kept it to Lake Okeechobee and input as there. But yeah, there were a couple of rainfall events, and you start to see them in the data set.
So those could be providing runoff, freshwater runoff from the urbanized or agricultural watershed that's compounding the
Absolutely.
conditions with the circulation problems.
Yeah.
But do we know that the sea level rise has changed that bathtub effect or that circulation pattern? Isolate that from
The sea level rise certainly over this time period, right? We're talking a month, okay? There's zero sea level rise.
Yeah.
The tides are a meter or so, which is not horribly large, but significant enough here because we are very flat around here. So a meter going up and down makes a big difference for the hydraulics. Water flows downhill. When downhill is more, i.e. low tide, it flows faster. So high tide slows everything down, low tide speeds it up. And, millimeters per whatever rate we're at will affect us over time periods. I mean, at some point, a septic system in Florida is not going to do much good. We're not there yet, but for a physics standpoint. From a nutrient release standpoint, I think we're pretty much there.
I did want to just reiterate that on our coast here, that modulation, the pulsing of discharges has kept us from having that layering effect. So we get a little bit more circulation. So we learned that a long time ago.
We have such high volumes. I mean, we get the flows that he was talking about were high, 1,800. Here
It's a
we're
smaller system.
10,000. So we have a much bigger system, and we get much more of the water. But again, I think to speak to some of the complementary nature of the two talks, we don't know anything about the inputs that are coming in on the other side of S79. We have this great TMDL, but then we have this area between the locks that is constantly fluctuating in terms of rainfall and discharge and use. So at some point we need to address that.
I was going to ask you, what do we need in order to have this kind of analysis over here on our much larger system?
Well, we had some great news this morning. It's been in the works for a long time, but we are getting nutrient sensors on our instrumentation, not at all sites, but at 3 sites, hopefully 2 or 3 sites. So we'll put them in McIntyre Creek to look at those phytoplankton bloom dynamics, and we'll put them in Fort Myers to look at the blue-green algae. I think Ian and I were talking earlier, there are some other sensors that detect blue-greens that would probably benefit us in terms of looking at Fort Myers, especially over the warmer parts of the year.
Can you separate out the effects between rainwater in the estuary and what comes down the Caloosahatchee?
I would say probably we don't have the source. We don't have the sources that are as distinct on the East Coast. We have our sources S79 and above, which is of course watershed, rainfall, runoff, and the lake, and then we also have the tidal basin too, and then the ocean as an end member. I've looked at some of our data in the same way that Ian has. In some cases you can, especially in Tarpon Bay, you can tell distinct end members, the ocean versus Tarpon Bay. So if you start to increase the number of sensors in an area and look at circulation, but our system is much more linear, even though we're sort of getting up into Pine Island a little bit. So
I'm thinking of trying to find a solution. And if we're the problem instead of the lake, or how do you separate that out?
right.
So,
And part of my research is actually looking for better methodologies of distinguishing. Within the seed on pool, there's lots of potential tracers if you start subdividing it up. The question is always effectiveness and reasonably priced and get the data out. I mean, you can always go out and grab samples and spend hours in the lab, but again, then that's not particularly management oriented.
You got to do it.
Yeah, it's got to be
Like
simpler.
a thermometer.
Yep.
Eric, I think there's a question
Yeah.
here.
I just had a quick question, which I just wanted to summarize your thing in the following way, see if I got it right. I think that most of us, after reading the reports and seeing all the news about the large bloom on the East Coast, heard that it was phosphorus coming out of Lake Okeechobee that was causing it. I think
Correct.
that was kind of what we got. I think that's what the press more or less said.
Yep.
But what you've said is it's a misunderstanding of the effect of the lake on the system.
Yep.
Okay, the phosphorus was not coming from the lake, it was coming from the North Fork. Exactly why you don't know. I mean, how it got there you don't quite know. But
Well,
it's coming
it's
but
runoff and septic systems.
Right, but how much of it was runoff and how much was septic system? Septic is not clear. How much was agricultural and how much of it was residential? Wasn't
Correct.
clear in particular. Okay. But-
From this particular event.
this particular event. But the effect that the lake was having on it was, by changing the dynamics of the system, it was permitting encouraging that phosphorus to come from the North Fork and down the system and create the bloom.
Yeah.
Is that right?
Giving the phytoplankton longer.
Yes.
And if it's hanging around longer, it's doubling that many more times. So if it,
Very interesting.
yeah.
Very
So you got it.
Okay.
So that means I can go now.
We can give the talk next time.
Thank you.
Yeah.
So fundamentally, since we cannot control the amount of water that's coming down the two rivers because of the possibility of flooding, we're going to continue to have this problem as long as we've got all the water coming down both rivers. In other words, if a couple of tropical storms come right now, both of the rivers are going to be experiencing a lot of water going down.
True, but really high flows are going to flush the entire system, would be my guess. So
So for us, it just pushes the boundary of the bay out into the Gulf, and we start seeing effects in the Gulf.
Downstream.
But on the East Coast, their volume for the, it drops off very fast. Very large volumes of water in the Gulf Stream. It just gets carried. But here, the effects just sort of extend the boundaries. And I think I did want to just point out the city of Sanibel, and a lot of us have put together a report saying these are long-term solutions for the lake and the area around the lake for creating storage and treatment. So I encourage you to look at that document. Unfortunately, short term, I mean, there's 2 tropical waves coming off of Africa. There aren't many solutions.
Eric, related to that, do you want to talk about the lag effect on our coastal nutrient?
Yeah, so one of the things that's been difficult here, as I mentioned, as we sort of extend the boundary of the estuary into the Gulf, the disconnect between the flow and the loading and what we see biological response-wise sort of increases. So, I mean, what we initially saw with those diatom blooms are very much classic examples, but what we're seeing now with macroalgae in certain areas, we don't have a direct connection of these far field drivers like high flows. They're probably a combination of factors, including periods of high flows from the S79, but also more localized sources. So those lag effects that we don't understand can manifest themselves several months to several years, possibly later. As we saw in the 2005, we had a lot of hurricanes, a lot of high volume discharges from S79, and then we started seeing strandings of macroalgae on the beach that lasted for 18 months. So I think that's what you wanted me to comment on. And
That would be immediate leaks.
yeah.
So I'm going to say one thing here.
Please.
This community, this institution has been a leader in doing this. And we're just starting to see how effective it can be. I mean, so you guys should be proud of yourselves for supporting this as a really cool model of getting data like this. It's not beholden to the government. It's not beholden to research dollars. The community has stepped up and funded this on about to enter your 10th year. And there's not a hell of a lot of data out there that's got that continuity on this and this effectiveness. So I think this is an amazing. I use you guys all the time. I lead people to this website. I tell them about this all the time because this is a really remarkable effort. And it's an effort that we have to replicate around the world because the seas are rising. We are changing the environment.
Any other discussion? Thank you.
Good work, guys.
Thanks for being here.
I had a question about the census question. Come back, this was asked before. But there are censuses in RECON that are not in our system. And it sounds like some of those censuses probably should be there. Just, if nothing else, to start gathering data, to be ready to gather the data, to look for the correlations of the type people are working on it. So what can the community do? We'd love to have some guidance on what you might be doing, how to fund it.
Well, I think a lot of the things that we do to raise money is through staff-initiated proposals, and we go through and put equipment in proposals, and some of those work and are funded, and some don't. But in general, I think we should entertain the idea of upgrading what we have with additional instrumentation for the future. We'll give you some guidance. How about that?
Well, and I'll building the models, starting to use this data not just as reactive, but models allow us to move into the future and say, what can we do 2 days from now? So my localized is, if you knew that Jimmy Buffett played down the street, a free concert, and 2 days ago, that would be interesting, and you'd be annoyed that you just found out. If you knew he was playing right now, okay, maybe you could get there. But if you knew he was going to be there tomorrow night, you would change your schedule to get to the free concert. And that's the value of going from observations that are lagged from science through real-time data to models. You can react to models. If you don't like Jimmy Buffett, that's okay.
Thank you all for being here.
Everybody likes Jimmy Buffett.
Thank you.